So, Ariel, maybe you can introduce our keynote speaker.
Yeah, we're good in time, which means that no, we lined up a speaker that will raise many questions. So don't hesitate to ask him. So Filippo, please come and take the stage. I hope your slides will come up. Filippo is an engineer physicist to start with. Moved to biology. I think there are some of us here that made similar trajectories, and in the conceptual work that he does, he is still an engineer and a physicist, even though he applies this to biological challenges, I would say, at scale. So, indeed, without further ado, tell us what biofoundries can do to make this change, and we'll try to follow your footsteps.
Fantastic.
Thanks for being
with
us.
Thank you very much, Ariel, and thanks for the invite. It's an absolute pleasure to be here with you today. So I've got my brief. So I will try to sort of give you an idea of what we have been doing in my group, and I would say across the pond with collaborators, in fact, on the other side of the US, on the Pacific Ocean side of the US, to actually take some of the innovation, some of the basic science, basic research that we have been carrying out over the past decade, closer and closer to the market and to the patients. This is something I need to start with. So today I'm going to talk about companies and companies in which I have financial interests. So this is my disclosure. The specific one I'm going to talk about is GenCirc. I hold shares in this company, so just be mindful of, certainly don't use information I will disclose to make investment decisions. This is the agenda. So this is what I plan to cover in the next 20-30 minutes. What I would like to do first is to walk you through the process of sort of going beyond the approach we have taken to therapies over the past 50 years and starting thinking about what synthetic biology can actually do to get us closer to smarter, more precise, more patient-tailored therapies with genetic circuits. And then I will slightly sort of move my attention to thinking about cells as living therapeutics. So sort of a shift in the way we think about therapies from essentially small molecules or genes, replacing genes, to actually thinking about the cell as essentially a living robot that we can deploy in the human body to actually go and do something that is of interest to us. And this is something that I would like you to sort of focus on as I progress in my talk, because some of the things that I think we have been collectively focusing on in the past 20 years, certainly with cell therapies, that the medium that we use to actually deploy these therapies is mammalian cells. And that's actually not necessarily true. So what I will do with GenCirc and the synchronized lysis circuit, which I will start shortening with SLC from now on, is actually thinking about bacteria as agents, as essentially vehicles to deliver a therapy. And then I will reflect a bit on what's at the horizon, where I see the most exciting opportunities for all of us active in this field. So first of all, the way we have been thinking about therapeutics has certainly shifted with our ability to engineer genetic circuits in the cell. This is obviously a fundamental philosophical change in the way we think about essentially agents that are active in the human body, the ability to essentially look at a cell as a processing unit, something that takes inputs, is able to do some computation with that input and decide based on that input what to do, how to what to secrete, is certainly an innovation in the way we have been thinking about therapies, certainly over the past several decades. And this is something that yesterday we were discussing about what the real difference is between synthetic biology and genome engineering and genetic engineering in general. This picture summarizes what, for me, is the key difference. It's not one gene at a time, it's not sort of making point-wise modifications in the genome of a cell, it's thinking about what dynamical function that circuit actually implements. And how are we going to assemble those circuits so that that processing from input to output actually gives us a new function, something that the cell wasn't doing before. So this is obviously a paradigm shift in the way we think about cells and engineering cells for our own purposes. And obviously it is daunting, because obviously we haven't been we have only been doing this for the past 20 years, and only now we are starting seeing the benefits of having that added flexibility of actually having a cell that in real time is actually computing and acting on that new information. So it is daunting, but it also opens a wide array of possibilities, because now all of a sudden we are not just, our focus when we develop therapies or when we develop active agents is not small molecules anymore, is not sort of the standard chemical physical chemistry of molecules. Our design space is not just that anymore, it is essentially a computer. And it's a computer that is living, and it's a computer that is sustaining its life inside the human body, and it's a computer that sometimes is also replicating. And sometimes we can exploit the ability of that computer, living computer, to actually replicate to our own sort of advantage. So this is, again, a frame shift in the way we think about therapies. And while sort of it does bring complexity, because obviously we haven't been designing therapeutic agents before like this, thinking that, imagine if we thought that, I don't know, take your favorite medicine, like paracetamol or whatever, was actually keeping, sort of duplicating itself in our body. This is completely different compared to what we have been used so far, but it does offer opportunities that we need to learn to exploit. So this is a bit of a frame shift, but at the same time, it does give us the opportunity to start thinking about the engineering of therapeutic agents more like the way we would engineer a bridge, hopefully. So this is my very engineering-based sort of take on how we should actually be doing engineering biology or synthetic biology. And obviously here I need to put my hands up and admit that this is a very sort of mechanistic, probably reductionist approach to engineering biology. So for me, everything in the end boils down to being able to synthesize and distill the function that I want our cells, those living therapeutic agents, to actually do in the body, being able to formalize that very often with a mathematical model, and then being able to translate those dynamical specifications into essentially plasmid design, okay? This is obviously something that the foundries are uniquely positioned to do, because they are able to take essentially the user on a journey where, starting from those designs, and sometimes they will be able to actually help you in optimizing those designs. So not just take your plasmid maps and just get the parts ordered and assembled, but sometimes they will be able, in fact, very often, they're able to help you in optimizing those designs, and then build those designs with those robots that are so powerful, screen those constructs, those cells, and even more importantly, test them, so get a sense of whether they're working or not, and identifying the actionable information that will allow you and them together to actually feed back what worked and what didn't, so that that loop can start again, okay? Yesterday we were talking about the analogy between civil engineering and the way we build bridges and the way we build cells. In an ideal world, I don't think that anybody here is fooling themselves on thinking that in the short term, in the next 5-10 years, you will be able to go to a foundry and say, I want a cell that makes this bioproduct. I'm not making any sort of any promise here. I don't think that there will be a realistic path from where we are now to that individual cell in one loop. But what, in my opinion, having seen what the foundries have been up to over the past 10 years or so, what is going to happen is that in that learn branch of this design, build, test, learn cycle, we will be able to optimize our design of experiments algorithm, so we will be able to make more and more use and leverage more and more the information that comes from the phenotyping in a way that maybe is gonna take 3 or 4 cycles of this sort of DBTL loop, but it's certainly gonna take a shorter and shorter amount of time, which in the end is what matters, because I'm sure most of you will agree with me, what matters is that what we find in our labs has the maximum impact possible on society, on our ultimate stakeholders, the people that in the end pay our salaries. But so I suspect that what's going to happen is that as foundries become more and more proficient in traveling through this DBTL cycle, we will see a shortening of essentially the characteristic time scale it takes to actually get to a point where you have your candidate therapeutics, for example. So this is something that, so this is a video. I'm hoping that the audio will work, but this is something that obviously in my lab with my group we have been particularly keen to explore, and in particular, how do we automate the process of designing those circuits based on specifications, based on essentially how we want those circuits to behave. And this is something that from sort of the bit of engineering that I am from, which is computer engineering, is actually a solved problem in consumer electronics. The reason why we have phones and computers essentially around us is that there is a thing that is called electronic design automation that practically allows algorithms to design the circuits for us. There is practically nobody in Samsung or Apple or none of the system integrators that actually designs circuits at low level. There are algorithms that do that. Those engineers, they set the specifications for those algorithms, for essentially the functions that those circuits have to carry out, and automatically they get essentially the blueprint of the circuit, okay? So a number of years ago now, we asked ourselves, can we do this with what we know of genetic circuits, of the dynamical behavior of parts? And this is our response. Hopefully, might not work. I will comment it in real time. Essentially, what the question is, can we actually think of our circuits, the circuits inside the cells, as electronic circuits? And can we use electronic design automation, so the way essentially an engineer would do this, and translate it to actual biological circuits? So can we actually use the models of those parts in order to automatically assemble them? This is obviously, this would unlock terrific innovations, because all of a sudden we can just say, I want this circuit to output this when it senses this signal in the microenvironment of the cell. And essentially what we have done has been to combine a standard ordinary differential equation approach, or literally like modeling the dynamics of circuits, with setting specifications on the envelope that those circuits, the dynamical behavior of that envelope that those circuits we sort of were designing, we wanted to have. And then we told essentially what is called a biodesign automation algorithm, which is essentially just a fancy word to say an optimizer, a piece of artificial intelligence, to go away and find all the combinations of those parts that were compatible, whose dynamical behavior was compatible with what we were expecting, okay? It's essentially a massive optimization exercise that is solved by an algorithm, not by humans. And essentially what happens is that allows us to explore more efficiently the design space, and in turn, this allows us to minimize the number of times that we have to go through the sample test learn, okay? So this is our response to how do we sort of help the robots in the foundry help us. So, fantastic. So we have some, and these algorithms, I mean, my group is not the only one that has been working on biodesign automation. There's many groups in the world, in fact, in France as well, that have been working on this. The question is, what can we do with these algorithms? Once we have this superpower, where we can say, I want to sort of be able to flick my fingers and get a circuit that performs this specific function, endows a cell with this new function, what can we build? This is from a review from last year on essentially a taxonomy of the sort of contributions of applications of synthetic biology in the space of health. And you can see it essentially from drug discovery to designing cancer cell therapies. We did hear about CAR T cells yesterday. The field is really maturing to a point where we are now able to realistically say, this is a disease, a disease accounting for this amount of global disease burden. Why don't we go after the disruption in the mechanics, in the dynamics of those cells in a way that actually looks like an engineering exercise rather than needing to build 50 bridges to get one, okay? So this is a very active area of research and something that is certainly promising. Now, I'm going to switch gears now, because so far, obviously, I've been discussing about very sort of high-level topics. What I was hoping, given the innovation angle of today, what I was hoping to do today was actually to talk about a real-world example of this process that turned into a startup called GenCirc. Now, GenCirc is a startup from Jeff Hastie's lab, one of the founding fathers of synthetic biology. And Jeff, a number of years ago, this is actually a joint work between Jeff's lab and Tal Danino's lab at Columbia University in New York City. So a number of years ago, in fact, back when I was a visiting PhD student in his lab, Jeff had this idea of a synchronized lyse circuit, essentially a very, very simple circuit in Escherichia coli, where you have a competing self-positive and self-negative feedback loop. So you have the LUXI, LUX inducer, that is essentially producing something that upregulates itself, and at the same time secretes, so the cell will secrete a quorum sensing molecule that will eventually trigger the expression of a Phi gene, so that lysis gene that you see in red. Essentially what happens is that the cell will produce something that will keep activating itself and will send signals to other cells to make them know that the density of cells around it, obviously if the cells are growing, will increase up to the point where there will be enough of the activator for the lysis gene that will immediately lyse the cells. And this makes for a beautiful sort of progress of essentially periodic spikes of lysis. Okay, so what happens is that cells will grow, will keep growing. Then at some point there will be a quorum threshold reached, and that's when you start seeing cells turning green. And after a few minutes of that quorum threshold being reached, cells will literally explode. Okay? Now, this was demonstrated in a paper back in 2016, and immediately, this is in fact the video. So you see, at some point, cells will just fire and then die. Grow, fire, and die. And this circuit works incredibly robustly, and this was sort of the key enabler of this innovation, because there's despite the fact that the cells that die are the result of a stochastic process, the percentage of cells that will survive the lysis event is actually fairly predictable. It's between 5 and 10%. So you see that essentially the number of cells that will remain to essentially seed the next population is fairly constant. Okay, fantastic. You can actually track these circuits and in a very consistent way. So there is no damping of the effect. There is no dependence of the amount of time that it takes between two lysis events that essentially depends on, for example, temperature. So a period between two lysis events is fairly independent of temperature. Even more interesting, and this is where I think the next wave of living therapeutics will leverage the advancements in on the modeling side. You can actually go back to the 2016 paper, and you will see a beautiful amount of work done on using modeling to understand the extent of the robustness of the circuit. So there's an accompanying model that Jeff's group put together to actually study, for example, where the oscillatory behavior would have been seen given the amount of flow around the cells, given essentially a degradation tag that they use and the strength of that. Bottom line, there was no way to actually kill those lysis events. So essentially, you would keep observing these growth, fire, lysis, growth, fire, lysis practically for weeks. Okay, which is great because this is actually empty field for releasing something in the body. So you do have these bacteria that keep growing, hit a threshold, lyse, keep growing, hit a threshold, lyse, and whenever they lyse, they can actually release a bioactive agent, so something that is actually doing something in the body. And the question was what? The obvious candidates were essentially anti-cancer drugs. So there is, in fact, for over a century, and mostly from work that happened in this country, we have known that very many strains of E. coli and in fact probiotic strains of bacteria actually have an innate chemotaxis, show an innate chemotaxis to microoxic environments, and tumor cores happen to be environments. The necrotic core of a tumor happens to be a microoxic environment. So for a number of, in fact, centuries, I would say, I think the first reports date back to the 1700s or 1800s, we have known that surgeons have essentially opened up tumors and found bacteria in them. So the first sort of immediate idea that Jeff had was to actually exploit the fact that bacteria could colonize that environment, and bacteria engineered with the SLC, with the synchronized lysis circuit, could actually continuously deliver very potent anti-cancer drugs that would otherwise not be a candidate for systemic administration because they were too poisonous in a synchronized way and in a very localized sort of fashion, because cells will only lyse when they feel essentially that there is enough density of other cells around them, because obviously they exploit quorum sensing. So this was the idea. Essentially, let's equip these bacteria, for example, with anti-CD47 nanobodies. So essentially, let's make them make this sort of payload. So let's equip them with this payload so that whenever they lyse, they release in the tumor microenvironment that anti-cancer drug. I wasn't expecting this, but I will tell you what this slide is supposed to tell you. Essentially, the question there is, obviously, what do you engineer this bacteria with? And the answer is pretty much anything that doesn't require post-translational machinery that would require essentially a eukaryotic cell. So you can use nanobodies, you can use chemokines, you can use cytokines, you can use toxins like alpha emolysing, which is actually the first example that I will show you. Essentially, what you can do is you can make these bacteria that will be sort of kamikazes in the tumor, make anything that will make the life of those cells in the tumor as complicated as possible. So this is actually, these are results from an in vivo study that was published back in 2016, where the team was able to essentially take an A20 lymphoma mouse model and essentially grow a tumor, and then treat those mice with either the SLC equipped with the anti-CD47 antibody, or SLC unarmed, what we would call the unarmed version of the SLC, and obviously compare it to just injecting PBS. So what happens here is that SLC can be delivered either intratumorally through oral route or directly in the bloodstream through the tail vein. In this case, the administration is intratumoral, and what they are looking at is, for example, tumor volume, so essentially how big the tumor is, over a function of time after the treatment with the 5 different treatments that you see there. So control SLC, so pure SLC, the monoclonal antibody, the monoclonal nanobody of CD47, or SLC plus CD47, the anti-CD47, and you can see that essentially, obviously, what you would expect is that there is an effect of the administration of the CD47 antibody or the nanobody, and you can actually observe that the growth of the tumor is actually not as fast as in the case of essentially of the control. But as you can see here, and this was absolutely key, this was something that when we realized we had this asset pitching to VCs, we call this the money slide, because I'm not sure whether I can actually point here. I can't. Fantastic. That's absolutely fine. So you can see that essentially our fully armed SLC is the green line on the top right of the screen, and you see that the tumor volume is actually going down as a function of time, and that's the only case in our treatments where that happens. So this was, I could literally tell you, and obviously we started pitching to biotech VCs, their face completely changed when they saw this graph. That was actually part of the Nature paper that was originally published in 2016. we also show, well, they also showed, I should say, that the SLC was, so the bacteria equipped with this circuit were not escaping the tumor once injected. Obviously, they also studied the survival of mice being challenged with tumors and treated with the circuit or control, so the circuit unarmed. And also, and this was something, again, this was the second wow moment in those pitches to VCs. Bottom right plot, you can see that they are plotting the tumor volume as a function of time. There's a first challenge at day 0, and then there's a second challenge at day 100. Now, the interesting thing there is that when the mouse is re-challenged with a tumor, there are dormant cells there that haven't escaped that microenvironment that will essentially reactivate, because the conditions for those cells to grow will be reestablished, that essentially keep the tumor volume to 0. Practically, they can't re-give a tumor to the mouse, because there is this almost like bacterial immunity that is there to fight the tumor growth. This was, so the two plots on the right, you could absolutely tell the change in the stare that investors had when they saw this, because they realized that there was an obvious opportunity here to invest. This is going to get even more complicated. Apologies, I didn't realize that there was an issue with the rendering. I will tell you what these pieces of evidence did for us. So essentially, there was obviously a massive concern. You can imagine these are mice, but eventually the objective was to move to, and still is, to move to patients. And then the question was, okay, you are giving patients that are cancer patients bacteria. What's that gonna do to somebody that might have a suppressed immune system, and what's gonna happen to those bacteria that sort of have reached that tumor? And you can see that in the top left plot, you can see that bacteria can only be found in the tumor area where they were, in this case, again, IT injected, and nowhere else. So not in the other flank where there was another tumor injected, and not in the liver, not in the spleen. And then there was, you could actually tell that this was something that worked above and beyond what other bacterial cancer therapies had demonstrated, where you can see that in the bottom left plot, other competitors hadn't been able to demonstrate that bacteria would have remained localized to the site where they were injected. So this was a USP for Gencerc, and still is. This is something that obviously gave investors comfort that we were essentially not injecting patients with something that would have killed them. And this is what, so out of sort of the results that Jeff's group pioneered, an entire field of cancer, bacterial cancer therapies sort of was established, that now has its own legitimacy, and it's something that, it's, to be honest, I mean, obviously, again, I am biased because I'm part of the company, but it is one of the most exciting areas of synthetic biology, and something that holds incredible promise, because the reality is that we've only just started scratching the surface of what this bacteria can do and can make in a localized, synchronized way. So I would like to finish with a couple of reflections on what awaits us, what we should expect. We were discussing about this yesterday in the roundtable. I do believe that there is a huge opportunity for this community to join forces with what is happening on the language modeling side in computer science, to actually start exploiting the power of those tools, large language models like ChatGPT and so on and so forth, to actually design genetic circuits in a way that is much more context aware and sort of considers the genomic context in which we are designing these circuits than we have done ever before. This is an article that if you haven't come across it, I would definitely encourage you to have a look at. This is Eric Schmidt, former CEO of Google, on the time towards the end of April this year, in what I think is essentially the manifesto for synthetic biology, for synthetic biology plus AI in the years to come. We need to be ready for biotech's ChatGPT moment. You might have heard the Astera Institute, ARC Institute have come up with these large language models to design essentially proteins from scratch. There is a CRISPR large language model. This is the time to join forces and scope what is possible in that space, because the reality is that we now understand that the ability of large language models to take into account contextual information about, for example, what surrounds our circuits in the genome of bacteria is something that humans can't grasp. I do think that there is something that this community, and obviously this is again a bit of food for thought for us to reflect on. There is a challenge that our community has that people that were working on ChatGPT didn't have, and that challenge is data. When OpenAI worked on GPT 3.5, or in fact GPT-2 and the predecessor of that, they had literally an entire internet to train those algorithms on. The best source of data in biology, what essentially granted the Nobel Prize for Chemistry this year, is PDB, okay? And the reality is that there is nothing that is even comparable in terms of quality and quantity for us. And the question is, where is the data that we need in order to train those models that we need in order to design genomes? Where is that going to come from? Foundries are obviously sort of in a unique position to fill that gap, but that requires planning. This is an article from on the Tech Review, on the MIT Tech Review, where essentially David Baker, one of the three researchers that shared the Nobel Prize for Chemistry with the AlphaFold, so Demis Hassabis and John Cooper, where they essentially reflect exactly on this. Our community, we can be the makers of our own fortunes, but we need to plan for it. And this is something that I don't think will be solved in the short term. We certainly have to devise better ways to acquire those data, to start data acquisition campaigns that then make the data available to the community so that we can have our own ChatGPT moment in biotech as well. Fantastic. So just a few last thoughts. I wanted to take just a couple of seconds to reflect on the incredible progress that this community achieved over the past 2 decades. We literally went from the first demonstrations, the repressilator, the toggle switch, all the way to being able to inject patients with cells that are engineered to go and attack cancer cells, which is absolutely mind-boggling. And there is something that I don't think we talk about enough, which is there is virtually no limit to the applications of those technologies. In fact, the limit is only set by our own imagination. And I think this bacterial cancer therapies is something that sort of speaks to that. The amount of, I wouldn't even say creativity, but how surprising is it that we can come up with something that is going after cancer cells that is actually a bacterium, so something that we would usually not want anywhere that is essentially not our gut, is crazy. And yet these therapies are having an impact on innovation and hopefully on patients today and in the near future. The one thing that I think we need to sort of take stock of and certainly use as a prompt to do more, and to, in my opinion, use as an excuse to go and talk to those communities at the boundary between computer science and biology. Certainly, those are those of us that are old enough to remember the bioinformatics sort of units in our centers. Those are excellent sort of conversation starters or places to have conversations. Biology and biotechnology have certainly under-leveraged or benefited less, sort of to a lesser extent than what should have, of architectures like transformers or post-transformers. And this is something that I see as, again, something that we can exploit and therefore a direction to go towards. In France, you have a unique context that I don't think is replicable in any other country in the EU and not in the UK either, which is you have probably two of the most exciting AI companies in the world, Mistral AI and Hugging Face. So I would imagine that your position here to exploit those connections is incredible. So just go and think creatively and think sort of proactively about establishing those connections. I will leave you with two predictions. The first one is that, in my opinion, the key challenge for this community will remain for a while, certainly in the short to medium term, coming up with predictable designs. Being able to, we haven't solved the biodesign automation problem in synthetic biology. We are getting there, but that the ability to design something on paper, nobody does. They design something in Benchling or in Genius, whatever you use, and then being able to actually come up with a vial of cells that do exactly what you thought they would, will remain an open challenge for a while. And the question is, what are we doing about it? How are we making sure that that challenge becomes so small that it's practically irrelevant for our research operations? My second prediction, something that I've anticipated already, is that I believe that, and this is connected to prediction one, I believe that for the foreseeable future, so even longer term than medium time frame, good quality, and that is the key there, good quality data, access to computational resources and automation will become the differentiator that will set the difference between success and sort of world-leading efforts and all the rest. So if I were a policymaker or a decision maker, I would certainly consider those areas where investment will actually give us competitive advantage. And I do believe that the future battles, when it will come to sort of being able to tame the complexity of biology, will be fought on those three fields. With that, I will stop and I will be more than happy to take any question you might have. Thank you.
Questions? You can vote or just start the mic and and start asking questions. Jesus, go.
I can probably hear you. I will repeat the question. Yeah. No, well, so the bacteria will be essentially under detection limit. So there will be small, yeah. 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 Yes. Yes. They remain, so the same site gets re-challenged with a tumor. So what happens is that the question is, I should have repeated the question. So if the tumor is essentially, if the mice is essentially re-given a tumor and the tumor doesn't grow, where are the bacteria? And obviously the answer is in the same place. So they remain in the same site, and whenever the conditions, essentially the microtoxic conditions, recreate, they will obviously regrow. Correct. Yes. Yeah, yeah, yeah. So it's the same tumor. Yeah, it's the same site.
More questions? So just a naive question: how do you avoid the immunogenicity of the bacteria? It's simply because you inject them directly to the tumor.
Yeah. So this is again IT. So it's literally like there is a. In fact, you can see it in the plot. There are massive efforts to use oral and in fact intravenous routes. In this case, this is actually E. coli Nissl, which is actually a probiotic. So there is no sort of, so it's a probiotic bacterium, but there is obviously, as you can imagine, there is a significant amount of interest to move away, or not move away, but explore other bacteria.
So where are we in sort of the optimal bacterial chassis?
Yeah.
For human treatment?
So the short answer is we are in sort of prehistoric age in this space. What the field, and this is part of the regulatory conversations that we were having, the reason why bacterial cancer therapies have focused on essentially wild type sort of bugs is that is the most obvious way you would use if you want to actually get to the market within your lifetime. Because otherwise, the question is, how do you get the chassis approved first? And you know that there's safety in the first place of the chassis before you even start like manipulating it.
But could you deliver your system to bacteria that are already there rather than putting bacteria inside?
I don't see why you couldn't. We obviously haven't done that. But there is, obviously what you're interested in is that the dynamics of the lysis are conserved and sort of robust to some extent. We have shown that, at least for bacteria, you can see it's a matter of, so they need to be there for a matter of weeks rather than months. So even if you had some kind of dumping effect of the actual lysis after a while, you wouldn't really care. So having a sort of an exogenous way to access those bacteria.
Or I mean, just infecting like
exactly.
like a red, like our bacteria route is phages, for example.
Yeah,
right?
exactly.
So you get your
maybe
system
the next
inside
gen
the
would be
cells
with phages, and to be honest, that would actually solve a number of headaches that I think all bacterial cancer therapies companies have.
I have a very different question. I very much like your conclusion, and because I'm already feeling that as a just standard researcher myself, you feel overwhelmed by data management. And I was wondering if you, as the head of a biofoundry, you were aware of basically a new field of data management science that is developing that could help us to think more smartly how to organize our research. Because I think that we are learning as we walk, and it's probably not the best way to do it.
Absolutely. So, yes, I am aware, so there's an effort which is actually called BioRDM. I will get back to you with sort of the link. So, which is actually biological research data management. Is an initiative that in the UK essentially handles the standardization of the essentially the
Type of data.
yeah, exactly. So if I want to publish time-lapse microscopy, for example, data, there is I can get in touch with them to make sure that the format of the files complies with fair regulations, but also or guidelines, but also that there is persistency and accessibility of the data. So yes, this is something
It's a new
huge.
field, yeah.
Yes. Also, keep like something that, and this is a absolutely brilliant question. Whenever we don't think about those things, we're leaving money on the table, because it means that we can't use those data to train those models or just advance the field. But also at the same time, we are losing track of, like, one of the issues that we have, for example, in my group, is that whenever a PhD student leaves, the only thing that or postdoc leaves, what we are left with are the plots of the paper and that needs to stop, because that's literally taxpayers' money that gets squandered. So yes, so BioRDM is the sort of the authority, at least in the UK, is the most sort of prominent one.
Thank you.
And can I just say on this, because I'm a particularly bullish about this. There are some organizations, there are some companies that have been able to actually make business models out of sort of data access. Again, Hugging Face, they have probably the biggest supervised fine-tuning sort of database of data sets. I wonder why we can't have the same thing. Why, if I have to, obviously we have Geo, we have Omnibus, we have the sort of very old school sort of bioinformatics stuff, deep sequencing and the sort of gene expression. But the reality is that if I want to do supervised fine-tuning or DPO on a large language model, it literally takes 5 lines of Python code to do that. And in biology, we don't have that. If I want to get the data from a paper of yours, I need to literally email you. And I mean, unless it's hoping, exactly, right? So it is crazy, and it doesn't require sort of, this is not against our values. It's just that we need to sort it out. We need to create infrastructure that allows us to share the data and use it almost seamlessly.
So, I mean, Ginkgo built an LLM, right, based on their data, and they sort of released it now. Is this a sort of a format that you think we should all adopt, or we should try to have our own system? So,
absolutely kudos to Ginkgo so you can obviously use it on their platform, obviously paying. So my question, yes, absolutely real, but we should strive as a community to allow that, but without being locked in their platform, right? As much as I absolutely love Ginkgo, the reality is that Ginkgo is using public data, right?
Our data.
So they certainly invested in the infrastructure. They have their own computational infrastructure, but I shouldn't have to pay to use their expression model. See what I mean? So yeah, it's time that we actually champion this.
Yes. Any other questions? So thanks, Filippo. Time for coffee.
My pleasure.
Meet here at 11:30.